I'm Daniel Levine, and this is the Bio Report. One of the challenges of treating brain tumors is delivering potent biologic therapies across the blood-brain barrier. Adaptin Bio has developed platform technology that harnesses a patient's own T cells to transport bispecific therapeutic payloads across the blood-brain barrier and into other targeted tissue, with an initial focus on treating glioblastoma. We spoke to Michael Roberts, co-founder and CEO of Adaptin Bio, about the unmet need in glioblastoma, the limitations of current blood-brain barrier crossing strategies, and how the company's platform seeks to change the treatment paradigm by using patient-derived T cells as delivery vehicles for targeted biologics. Michael, thanks for joining us.
It's a pleasure to be here, Danny.
We're going to talk about difficult-to-treat brain tumors, Adaptin Bio, and its platform technology for developing therapies across the blood-brain barrier. Let's start with glioblastoma. What is it?
Glioblastoma, it's often called glioblastoma multiforme, or GBM for short. And it's a grade 4 astrocytoma, which means it's the highest stage of malignancy, meaning that it's by the time you're already diagnosed, it's well advanced. So it is the most aggressive of the primary brain tumors in adults, and it's characterized by a couple of things. First, it's rapid growth, right? So once it's diagnosed, we're talking rapid growth within months. It has what's called diffuse infiltration, which means that it goes into the surrounding brain tissue. I like to tell people that if they just put their hand up and they spread their fingers, what the physician sees on the radiographic image is basically the palm of your hand. That's what they can see. The fingers represent tentacles of tumor cells that go into the rest of the brain that they don't see, right? So it has this diffuse infiltration. And then, of course, I think one of the most important things here is it has a marked genetic and cellular heterogeneity, meaning that not all cells in the tumor are alike. They have very different behaviors. They have different signatures. So when you treat the brain tumor, it's with a single entity, it's going to escape. It's not going to be effective. Okay. So one thing I can tell about glioblastoma is that it is relentless, right? So it quickly develops by the time the patient gets to the physician, and typically some of the symptoms of the patients are, persistent and worsening headaches. They get seizures, which is often the first thing, because again, if you have headaches all the time, that may not be a brain tumor, but when you have a seizure, that usually tells you that there's something else involved. And then, of course, family, friends, they may see cognitive or personality changes in you. And then there's these things called focal neurological episodes. And these are things like weaknesses in your hand, visual changes, speech problems. Those things rapidly come up too. And of course, as you have a growing tumor in the brain, it increases the intracranial pressure. And that causes things like nausea and vomiting. So those are some of the symptoms, and they develop extremely quickly, over weeks and even months. So it just is, it's relentless, it's quick, and obviously there's a huge unmet need here.
And why is it tend to get diagnosed so late? Is it that the early symptoms are just something that might seem innocuous, or is it the rapidity with which it advances?
Right. So, I mean, everybody gets headaches once in a while, right? It's just when it becomes persistent and worsens over a period of time. Again, it can be months. And And so, the initial symptoms are very, they're not noticeable as abnormal, right? And then by the time um, somebody kind of believes that something's wrong, it's way advanced, right? Glioblastoma is classified as a grade 4. It's not grade 3. That's not glioblastoma. It's grade 4. So it is already advanced to the most, latest stage of tumor as you can get.
And how effective are current therapies, and how large an unmet need does this represent?
Right. So obviously, there's a lot of challenges to treating the disease. They're not very effective. So over the last 20 years, there's been basically no advancement in the clinical outcome of various treatments, right? And the reason being is that, as I mentioned before, glioblastoma is heterogeneous. When you target a single pathway, for example, by a small molecule drug or something, not all cells have that. So it is resistant to certain mechanisms. So therefore it grows rapidly, right? The other thing is that Mother Nature put in a thing called the blood-brain barrier, right? So the vasculature in the brain is significantly different from that's within the rest part of the body. For the reason that the brain is a very highly sensitive organ, and you don't want pathogens and toxins to enter. So Mother Nature did a really smart thing here and developed this blood-brain barrier to prevent that, right? So the blood-brain barrier is tightly packed endothelial cells, the lining of the blood vessels, and there are other aspects which prevent passage of molecules into the brain. So it prevents those things, which is great. But the problem in treatment is that it also blocks drugs. So we can't get it into the brain. And therefore, these two things, particularly with glioblastoma, are the major hurdles and why, there's been limited to no success in treating this disease. And, as I mentioned before, it's rapidly progressing, right? So if you do take it out via, standard of care, which is actually tumor resection first, then you go through radiotherapy, and then you go through chemotherapy, right? Even within that, patients only survive typically less than 24 months. And once recurrence occurs, survival's only about 8 months. So it's just relentless. It just keeps coming back. And of course, the problem with treating it is you can't get things into the brain sufficiently.
There are strategies for getting therapeutic payloads across the blood-brain barrier. What are the limitations of existing approaches?
Right. So they're, significant. So there are a couple of things that you can do for getting things into the brain, right? There's the direct application, right? You can do injections, for example, in glioblastoma, you can do direct injections, intratumoral injections. You can deliver drugs via what's called a convection-enhanced delivery, which is a technique to get things past the blood-brain barrier. You can provide drugs intrathecally, but these are really always invasive, right? So you prefer not to do surgical procedures. With these, they give drugs locally. As glioblastoma is very diffuse, it doesn't target all the tumor cells. Therefore, it's very localized. And of course, there's other issues with direct injection. And then there's other techniques, things which actually are coming much more popular now. One is focused ultrasound with these things called microbubbles. And what they do is they give injections of these little tiny bubbles in the bloodstream. And then they take ultrasound and they pulse it. And what happens is those microbubbles cavitate and they open up this blood-brain barrier to drugs potentially that are administered, right? But the problem there is that it opens it up to everything, right? Toxins, things like that. And it's not really extremely controllable. And of course, it's localized, right? You can only do this at a certain area, right? So it doesn't encompass the all diffuse nature of the tumor. Some other things that you can use is receptor-mediated transcytosis, right? So the endothelial cells on the blood vessels have certain receptors that transport particular molecules into the brain, like glucose, insulin, transferrin, things like that, where the brain needs these nutrients to survive. So it has receptors to transport those in. So what you can do is hijack those transcytosis mechanisms. For example, there's a lot of work going on now with transferrin receptor and attaching beta amyloid antibodies to that so that it transports those drugs into the brain to treat Alzheimer's disease. And you're seeing a much better transport of these and much higher concentrations of large proteins into the brain. So those are techniques. The problem with glioblastoma in particular, by just delivering therapies, it's not individual therapies. It's not sufficient. As I said, these tumor cells change over time. They're relentless, and you have to adapt. So by just delivering a single molecule is not going to be sufficient. And then there's other things, nanoparticles, liposomes to transport small molecules into the brain more effectively because of half-life and clearance mechanisms. Those give the ability to transport into the brain over a longer period of time and can be more effective. And then lastly is, cell-based delivery, which is part of the technology that Adaptin is pursuing with a little bit of, modifications.
Adaptin Bio has platform technology to address the challenge of delivering therapies across the blood-brain barrier. This grew out of research at Duke University. What is the BRIGHT platform and how does it work?
Right. So the BRIGHT platform is an acronym for Brain Bispecific T-Cell Engager. And what it is a two-component system. One is a delivery vehicle, which we use the human immune system as that vehicle, right? So the immune system has the ability to transport or travel, basically, to any part of the body. And so it's a natural delivery vehicle. The other part of the BRIGHT platform is what we call a T-cell engager. This is a protein, a bispecific protein that targets both a tumor cell or tumor cell antigen, and it has a antibody to a T-cell receptor. For example, what's called a CD3 receptor. So it's a bispecific protein, and we use the delivery vehicle, the immune cell, the T cell, to transport that T cell engager into the brain. So what we do is we take from the patient their T cells via a process called leukapheresis, which is standard on all hospitals around the country. And you take it back to the lab, you isolate the T cells, we manipulate these T cells to express certain receptors that allow it to transport into the brain via passage by the blood-brain barrier. We take these T cell engagers, we can link them to that T cell delivery vehicle via the CD3 receptor, which then hijacks or transports that T cell engager into the brain. And then when both get into the brain, the T cell engager binds to the tumor cell and it brings in that T cell. And when you bring the two together, it activates that immune cell or the T cell to specifically kill that tumor cell via what's called a perforin granzyme B pathway. And so this is highly specific. And the importance of this is that in particular, in glioblastoma, which is a tumor, which is what we call cold, right? So it doesn't express a lot of antigen. It's not inflammatory, so there's not a lot of T cells or immune cells that are present. So our delivery vehicle is actually part of the therapeutic. If we not have the delivery vehicle in the brain at the tumor microenvironment, the T cell engager alone would not likely be highly effective. And so you can't give just the T cell engager or just the T cell or the delivery vehicle. You have to combine the two so that it can be more effective.
And just to clarify, this is a very specific transport technology that's designed to get the engineered T cells across the blood-brain barrier as opposed to carrying broader payloads.
That's right. So again, the Bright platform is specific to a T cell engager, right? So that's why it's called Bright. But the technology is much, much broader than that, right? So the T cells that we isolate, and we don't genetically engineer them. We manipulate them in a way that is scalable, that's relatively quick. So we want to keep it simple. We want to be able to give this, technology and product to the broadest set of patients that we can. And then what we can do, we can take any drug. Doesn't necessarily need to be a T cell engager. It can be an antibody to a beta amyloid. It can be a oligonucleotide for a genetic, manipulation. But we can then attach or link those drugs to the carrier, the T cell, the immune cell, and transport it into the brain. So it's not just related to a T cell engager. It can be a broad set of therapeutic candidates ranging from, small molecules, oligonucleotides to proteins for a variety of indications. So it has broad applications just beyond glioblastoma.
And what advantages does this approach have?
So, and particularly with glioblastoma, it is very highly specific, right? So, glioblastoma is a cold tumor. It doesn't have many T cells present, so you're not going to fight the cancer. So, by giving that delivery vehicle of T cell, we can enhance the number of T cells in that tumor microenvironment, thereby being effective. Now, it creates what's called a hot tumor or inflammatory activity in the tumor microenvironment, which in tumor cells is what you would need and want. But say, for example, in Alzheimer's disease, in Alzheimer's, you don't want inflammation. That actually is part of the pathology of Alzheimer's. You don't want inflammation. And therefore, you can give other types of T cells called, say, regulatory T cells, which blunt that immune response. And therefore, you can deliver in Alzheimer's disease sort of a combination of beta amyloid clearance and a reduction of inflammation, thereby potentially improving the therapeutic effect of that drug. So we can change how we deliver these by the T cell that we use for a specific disease.
And what is the process like to create these therapies? How complicated is it to manufacture them?
Right. So we keep it really simple, right? Again, as I said, to be able to make it cost effective, being able to scale, and being able to give it to as many patients as we possibly can, we keep it very simple. So what we do is we take T cells from patients via, as I said before, a process called leukapheresis. It's a standard process. We isolate those T cells. We take it to our labs, particularly right now it's at Duke University, and we manipulate them, we activate them, we expand them. And so when you expand them, you're not just giving it potentially as a single dose. You can use that one expansion can represent, tens or hundreds of potential injections to transport a drug through the blood-brain barrier. So it's scalable, right? And of course, so we keep it very simple. And, the T cell engager, for example, is manufactured at a third-party CMO. And that is relatively inexpensive. If you were to do something that was genetically engineered, you'd have to have the cost and the, expense of say, a viral vector in order to genetically engineer a T cell. We don't do that. We're keeping it simple, which again can keep the cost down, make it quick. So that's what we're doing.
Your lead experimental therapy is APTN 101. What is this and how does it work?
Right. APTN 101 is our lead program that combines a T cell engager with our manipulated T cells. The T cell engager is a bispecific antibody where one end targets an antigen on tumor cell called EGFR-V3. That is a tumor-specific antigen. It does not, it's not on any other cell in the body other than tumors. So normal tissue, it's not present. So this is a very highly specific therapy. The other end is a CD3. So it binds to CD3 on the T cell. And again, that's the lead program. So what we do is we take these manipulated T cells, combine it with the T cell engager, which targets EGFR-V3. We administer both via IV infusion. We don't directly go into the brain, but we do IV infusion. The importance there is that you can get broad distribution throughout the brain, so you can target the largest area of tumors that are in the brain. So it's not just local to the main tumor mass. It can target other cells that are in the brain. And so we just administer both, and it transports it into the brain, giving you, highly specific and potentially highly effective tumor cell killing.
Why EGFR V3 and CD3?
Right. So EGFR-V3 is a highly specific antigen on tumor cells only. So it's highly specific, right? And it also demonstrates that a tumor cell has aggressive growth, right? So it's specific. It represents a tumor cell that has high growth, and therefore we want to target that. And the reason we target a very specific antigen is that it can limit any toxicity that's associated as what's called off-target toxicity. We can limit any toxicity while being very specific to the tumor cell. And of course, CD3 is the activation part of the T cell. When the CD3 bispecific antibody binds to the CD3 receptor complex on the T cell, it will activate that T cell and say, T cell, kill that tumor cell that's right by that, right? So it links the tumor cell with the T cell, and it specifically targets that tumor cell.
How is it administered?
Right. So it's administered via IV infusion, right? So we're not giving direct injection. It's IV infusion over basically a 4-day period. So we can maintain therapeutic concentrations because you want to maintain that for a period of time because of the half-life of the antibody is relatively quick. So we do an IV infusion over, say, a 4-day period. And what that allows is it to target the diffuse parts of the brain, right? If we were doing a local injection, it'd be very localized. But this gives us the ability to target, the broad parts of the other parts of the brain. Of course, we want to minimize any impact on patients. So therefore, IV infusion is a very common and fairly non-invasive procedure. So therefore, we give it via IV infusion.
And what's known about it from studies you've conducted to date?
Right. So we've got a wealth of preclinical, which is prior to getting into patients, studies where we have demonstrated that we can increase the distribution of large proteins like a T cell engager into the brain by more than 7-fold. So we can dramatically improve the crossing of these proteins into the brain. And our lead does it by more than 7-fold. We have other preclinical models which demonstrate the mechanism, right? So you put it in what we do as a gold standard glioblastoma tumor model, where you implant the tumor cells behind the blood-brain barrier into the brain, and then you administer, basically what's termed a placebo or the components of the active drug and the actual drug itself. And we've demonstrated in these preclinical models that we can have very robust tumor cell killing, which allow 70 to 80% of mice to be long-term survivors. So I don't like to use the word cure, but these animals become long-term survivors, which is at the end of the day is what we want to see in patients, right? So if these animal models, which sometimes don't, translate into human therapeutic effect, this will be a significant advancement in the treatment of glioblastoma. So we've got those studies. We have a wealth of safety studies, right? You do a panel of toxicity studies, which show that because it's specific to EGFRV3, there's no or very limited off-target toxicity here. So we can do this potentially in patients with low adverse event profile, right? So we've got a wealth of data, both efficacy as well as safety, that suggests that this could be, highly effective and low AE therapy for glioblastoma.
What's the development path forward?
So we currently have approval by FDA to initiate a phase 1 trial, which is a safety study. And we got approval to do that. We expect to be in our phase 1 trial at the early part of this year, likely the first quarter of 2026. And that phase 1 trial will study both recurrent and newly diagnosed GBM patients where they undergo dose escalation. So you start with low doses, and if you obviously have if there's no safety signals, then you increase dose. So we continue to increase dose until we reach either a maximal tolerable dose, or we see significant clinical effects, and we then stop there. So we are going to begin that study in the first quarter of this year.
You talked about the bright platforms applications beyond T cells, but how broadly are you potentially considering this for building a pipeline around?
It's so right now our primary target is primary tumors or glioblastoma, but it can be used obviously for other types of tumors in the brain. For example, brain metastases or what's called secondary tumors. For example, about 30% of all HER2 positive breast cancer patients die not because of their peripheral disease, but because of brain metastases. And therefore, we could potentially use this as a treatment for secondary tumors, right? Glioblastoma, there's about 15,000 patients diagnosed annually in the US. There's a larger population of lung cancer patients, breast cancer patients, melanoma patients that have secondary metastases in the brain. And therefore, that's a larger population that we could potentially treat. And then you can go beyond sort of the brain tumor aspects. So our focus would be other high unmet medical needs in the neurodegenerative space, things like Alzheimer's disease, Parkinson's disease, ALS, where we can deliver potentially antibodies and other therapies, oligonucleotides, into the brain to treat those types of diseases. Combining it with sort of an anti-inflammatory component. Right now, other treatments don't include that. We think that could be an additive effect, which we would have with our technology. So we can go into those neurodegenerative disorders. And then, of course, we have other genetic CNS diseases that we could potentially go into as well.
Given that, are you exploring the potential of licensing the technology or entering into co-development partnerships?
Sure. So the thing that we can control is our pipeline. And so we expect and we are building a CNS pipeline, which will start with our lead APTN 101 in primary brain tumors, but we're going to expand that and are expanding it into secondary tumors. And then, of course, beyond that, we'll go into other neurodegenerative diseases. But that's what we can control, right? So we can build a pipeline and plan to build a pipeline, and we'll initiate that internally. So we'll do the early stage development of that. But of course, as with any company, you want to be able to bring your drugs to market as quickly as possible. So therefore, strategic transactions like partnering can also happen. So we would be looking to other companies, large pharma, to take on our drugs and continue developing them in the clinic. We would also potentially combine our technology with their drugs. So improving upon what they're doing using our technology. So we've got multiple avenues of bringing our drugs to the market with, as quickly as we possibly can for as little money as we need to be able to do it via partnering. And so we plan to do that. At the end of the day, our goal is to make the most effective treatments for patients. And if we're successful at that, other things will, come in. We'll have companies interested in our products. We will commercialize them, get them to patients. So the thing that we can control is the products that we develop and be successful at developing those products. Everything else should fall in place.
The company completed a reverse merger in February. Can you explain the transaction and the associated financing?
Sure. We did what we call as a reverse merger with a public shell company, what's called a Form 10 company, the company being Unite Acquisition One Corporation. Um, People tend to think of them as like SPACs, but this was not a SPAC or a special purpose acquisition company. SPACs usually raise money in a process of an IPO. We merged with Unite One Acquisition as a Form 10, which does not um, raise money in an IPO. It's just a public reporting company. So, but with the intent of a merger, and so we merged with the Form 10 company, which allowed Adaptin to have now access to a bigger pool of potential investors. Right, are some investors only invest in public companies. And so now we have the opportunity to, you know, uh, have investors that are both public and private. But the transaction was completed in February, March of 2025. We raised about a total of $7.7 million dollars in that particular transaction. And so, that's basically part of that transaction. It's all public knowledge at this point.
The company is on the OTCQB. Why list there? Why not enjoy the greater freedom as a private company? Do you think you'll be able to better access capital you need?
Yeah. So we're not, as of today, listed on OTC. That is a process that we're undergoing right now. So when we're merging with a Form 10 company, it's not a public trading company. So it's just a reporting company. So we have to go through a process. And we're going through that process right now called a 211, where you get a ticker symbol and you start listing. And the first place you list is OTC. And so that's where we're in the process of doing. Um, the good thing about that, as I said, it brings in a larger population of potential investors, right? So it gives investors that we currently have, it gives new investors liquidity, right? So a lot of people like to be able to sell shares when it goes up, right? So that's the benefit of going with a public uh, you know, transaction. So it gives us a larger pool of investors, and it gives our investors the flexibility to get in and get out with based on their needs.
And how far will existing cash take you, and what's the plan for raising additional capital?
Right. So the existing cash will be suitable for getting into our phase one trial. So we will be needing additional capital in due course to complete our trials in our phase one trial and beyond. So this will be a continuous process, but yeah, the money is in order to get into our phase one trial, which will initiate in the first quarter of 2026.
And at this point, is there a plan for building a pipeline beyond 101?
Yeah, so we've got basically a 102, which is a specific antigen in tumor cells as well. We'll be using that in our primary tumors, so glioblastoma, for example, to enhance the effectiveness, if you will, of a changing tumor. So there's an entire process that I haven't described on the call here that our technology is very good at adapting. So we can change as the tumor changes. Glioblastoma is obviously relentless, but it changes. When there's therapeutic pressure, the tumor cells change their expression, they change the metabolism, things like that. Our technology is able to change as the tumor changes. So therefore, we can potentially be one step ahead of the tumor cell. Therefore, could potentially be effective. And 102 would be part of that kind of process to change as the tumor changes. Right now, no other technology has the ability to do that. And then, of course, we'll be working on additional treatments for neurodegenerative diseases. And so that's of interest to us as well.
As you think about balancing opportunities with the financing you have, how do you prioritize the indications you'll pursue?
Right. So it's somewhat based on the unmet need, right? So in GBM, for example, there's a huge unmet need. And can we be effective there, right? So if we think we have something that can be very effective with a high unmet need, we'll likely want to initiate that first. So that's our priority, is to use our technology in GBM to potentially have an effective therapy. Now, beyond that, it'll be based on, again, that unmet need. Are there other drugs that are currently approved in that indication, and how effective are they? And if there's limited effectiveness, then that's an area we'll go into, right? Or potentially go into. But what we also want to do is leverage the expertise we have. So, for example, if we're focusing on glioblastoma, we may have a higher priority for secondary tumors and developing a drug for a secondary tumor. So we want to also leverage the expertise and the resources that we have in those other therapeutic areas.
Michael Roberts, co-founder and CEO of Adaptin Bio. Michael, thanks so much for your time today.
Thank you, Daniel.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.